Hello and welcome to the 2023 Sexual Assault Awareness Month webinar series. Our third webinar of the four-part series is Touch DNA Evidence Collection in Sexual Assault Cases. Knowledge to Inform Practice. Our presenter today is Dr. Julie L. Valentine, Associate Dean and Associate Professor at the Brigham Young University College of Nursing, and also a Certified Sexual Assault Nurse Examiner with Wasatch Forensic Nurses. Without further delay, here is Dr. Valentine.
Welcome to the webinar on Touch DNA Evidence Collection in Sexual Assault Cases, Knowledge to Inform Practice. I would like to acknowledge support from this project from RTI International and Forensic Technology Center of Excellence. Also a thank you to Utah Bureau of Forensic Services and acknowledgement to my research colleagues and multiple research assistants. So let's dive in. So your learning objectives for this webinar, the end of this webinar, participants will be able to discuss factors that impact the likelihood of developing probative touch DNA profiles in sexual assault cases, verbalize understanding of best practices for evidence collection for touch DNA, sexual assault cases, and apply current research findings on touch DNA to inform multidisciplinary practice. To begin, we're going to start with a case study. This is a sexual assault case that happened on a campus that was a groping sexual assault case. So to frame this, Utah Bureau of Forensic Services, which is the state crime lab in Utah and serves the entire state, started adopting more advanced DNA testing measures in 2010, which included ID Plus and Y-Filer. We have a fabulous collaborative relationship between the State Crime Lab and the Sexual Assault Nurse Examiner teams. This is critically important because when you think about it, we work on the same stuff. Those that collect the evidence need to communicate and get feedback from those who analyze the evidence. I have presented in some jurisdictions where there's not great communication between the forensic medical examiners or sexual assault nurse examiners and the crime lab, and that really does not serve anyone well. There needs to be great communication. So as they move to these new testing measures, the forensic scientists were going to the sexual assault nurse examiner teams and presenting on these new testing measures. And it was at a meeting in early 2011 when one of the forensic scientists was presenting on this and sharing about the enhanced capabilities of these testing measures. And one of the sexual assault nurse examiners asked, does this mean that we should swab body areas where the suspect touched, like in stranger assaults? Because we were talking about the potential capabilities of developing DNA from epithelial cells or touch. And the forensic scientist said, I don't know, because we didn't know. We didn't know in practice what this might mean. All right, so let's fast forward a couple months. A sexual assault nurse examiner who was on call was paged to collect evidence on a victim who had been, she was told, groped on a college campus. But particularly, the SANE was told that they wanted evidence collected from the victim's mouth as she bit the suspect's finger. They wanted to have the SANE come to the law enforcement agency, the campus agency, rather than go to a hospital because they said, we just need you to swab her mouth. There's no injuries, and the victim did not want to go to the hospital where the exams usually take place. So the SANE said, okay, don't give her anything to eat or drink. I will be there in a half an hour. When the SANE arrived, she found it was a busy setting in this law enforcement agency and learned that they believe the same perpetrator had been hiding in a girl's locker room and stolen underwear and was watching girls undress and then was discovered and ran out of there. And that there were actually 3 additional groping victims in about an 8-hour time period all around this campus setting. So, as you can imagine, huge concern about what was happening. So the SANE went down to meet with this victim who was in a room to swab her mouth. Again, she'd been told that the victim had bit the finger of the perpetrator. And when the SANE opened the door, she found that the victim was eating a Subway sandwich and a drink. Those of you that know that those salivary enzymes, boy, they really eat up DNA. So she turned to the officer she was with and said, "She's eating. I don't think I can get anything from her mouth. Let me go in and talk with her." So the SANE, the forensic nurse, talked to the victim and said, "We're going to collect evidence where you tell me you have been touched," because the SANE had been at that meeting where the forensic scientists had said, "We don't know our capabilities about collecting touch DNA." So the SANE thought, this is probably all I've got because I'm probably not going to get anything from the oral cavity or her mouth. So she went through the history with the victim, and this was actually a very violent sexual assault. The victim reported that she had walked into a parking lot. At this point, the other assaults had happened in the daylight hours. This was wintertime. It was dark. She was walking to her car, and the perpetrator, really a large man, I think 6'5", 280 pounds. And this was a small, petite victim. As she was walking to her car, the perpetrator rounded the corner of another car. He was wearing a hoodie with a long trench coat. He started walking towards her. She immediately got very nervous. She pushed to unlock her car. When she got to the driver's side, and opened the door, he attacked her. He pushed her down over the driver's seat and the passenger seat. She was screaming and kicking. He put his hand over her mouth. That's how she was able to bite his finger to prevent her from screaming. His other hand, she was wearing a coat that one button was done. He tried to undo her button of her coat. He then put his other hand up her coat. She was wearing a skirt and tights, went under her skirt and down her tights and to her genital area. And his hand was on the front part of her underwear. So let's review where the victim or patient was touched. Oral cavity, finger went in the mouth, she bit the finger, but all around her face, that oral cavity. He grabbed her shoulders when he shoved her down across the seats around the jacket button, which was a smooth metal button, her abdomen, the tights, and the underwear. Now, you'll notice I have the kind of composition of those items here. Did you notice it was lacy fabric on the front part of the underwear, not smooth? And we're going to go back and talk about how that matters. So the forensic nurse moistened swabs with sterile water, collected, and non-moistened swab collected the oral, but also the lips around the mouth and chin, the lower abdomen, the jacket button, and the outside and front portion of the underwear. The SANE actually asked the victim to point to exactly where she thought she had been touched. So she had the guidance of the victim right there. The victim was also willing to provide her clothing. An advocate came and provided clothing for this victim to wear. So the SANE packaged up the clothing. Our current recommendations are for the forensic examiners not to collect swabs from the clothing, but just to collect the clothing if the patient or victim is willing to provide that clothing. Law enforcement on their end identified 5 suspects that matched the description of the assailant. One of these suspects had a misdemeanor charge about a year earlier for voyeurism on the campus and fit the description of a large man. And a couple days after this happened, they found this person at his place of employment and found that he had a Band-Aid on his finger that was a healing bite mark. They got a buccal swab from this suspect. So let's talk about what we found on the DNA yield. YSTR and STR were both done in this case. So on the upper part in the white are swabs collected by the forensic scientists in the crime lab, because remember, the clothes were collected and submitted. In the gray are swabs collected by the SANE at the time of the exam. So you'll see the skirt, no male DNA was found. The jacket shoulders, this was not collected by the SANE, but was found to have male DNA that was a mixture, but the suspect matched the major portion of that on the YSTR. Tights, also a mixture, but not as large a mixture, but the suspect matched the major portion of that. The underwear, they found no male DNA. When we went back and looked at this case in more detail, where do we usually collect on underwear? Where do we usually collect swabs? It's usually from the crotch. But remember, the victim had said that she was touched on the lacy front panel where his hand slid down onto the front part of her genitalia. So this underwear swab that was collected by the forensic scientist was from the crotch. Now the swabs collected by the SANE, the lower abdomen, YSTR, suspect matched a major portion. Underwear, that front lacy panel, also matched the major portion. Jacket button, no male DNA was found. The SANE had written in her notes that as they were doing the interview, the victim had actually put some lotion on her hands right before the SANE had gotten there, and she was fiddling with this smooth metal jacket button. Remember, smooth metal, and that she had been touching that. Well, they actually found no male DNA on the jacket button. Around her mouth and chin, yes, matched the major portion. So now the gold star, right? What we really want when we are testing is to get an STR profile that can then go into CODIS. Well, the forensic scientist did not develop an STR. There was not enough from their swabs DNA to develop an STR profile, but that swab that the SANE collected from this rough kind of lacy panel that was the front part of the underwear, a full STR DNA profile was developed that matched the suspect. This was huge. This was just from his hand, the epithelial cells. So the prosecution outcome. This victim was unable to identify the suspect in a photo lineup. This was due to the trauma from the event. This was a very successful student who, after this happened, needed to take some time off from school and for therapy and to heal, and was unable. And remember, this perpetrator had a hoodie on with the hood pulled up and a trench coat, but the DNA findings, the prosecutors stated, made all the difference. A plea bargain was reached. The suspect was sentenced to prison for up to 5 years. He did not serve that amount of time. He was released, and he has since been arrested 3 additional times for reoffending. The latest was just in the last couple of weeks. The victim outcome, which is so critically important in all of this. She was notified that her case and what we found that we were able to do with touch DNA in sexual assault cases really changed our practice in our state, and we're trying to change the practice nationally on this. And she said that knowing that has helped with her healing as much as or more than receiving the therapy, but she is now doing well. So this case changed practice, and we do have sometimes cases like this. In the state of Utah, we developed a touch DNA form. We changed our Utah sexual assault examination form, and we have really launched on this education path, which this webinar is part of, and I will tell you additional information that you can get on this. So now let's go into a little bit more on what does the literature tell us about touch DNA. And specifically, we think of touch DNA in groping sexual assaults. That's what we're really focused on, where we are not going to expect bodily fluids. And we also think about sexual assault cases by strangers. Because strangers were not expecting to find that person's DNA on the victim. We can also think about the importance of touch DNA when anytime foreign contributor DNA is not expected as background DNA, such as if there's a protective order and that person's DNA should not be on that victim, also can be helpful in those cases. We also can think about collecting touch DNA from a suspect of the victim as well. Touch DNA. So let's dive into when did this really start? Well, in 1997, Van Oorschot, who's done a bulk of or a large amount of the research that we know on touch DNA, had a small paper in Nature that talked about the idea of collecting DNA from fingerprints. So not just collecting the fingerprints, but what about getting the DNA from these epithelial cells? We didn't really have guidelines on this, especially in sexual assault cases, until the publication by the National Institute of Justice on the National Best Practices for Sexual Assault Kits, a multidisciplinary approach that was published in 2017. So now let's go into background on what we know from research about what impacts the amount of touch DNA left on an object. So we're going to talk about donor shedder status, because that matters a lot. We're going to talk about the surface of the object. We talked about that a little bit with this case study regarding smooth textures or the rough textures. And I talked about the lace of that underwear. We're also going to talk about the type of contact and the length of time between contact and evidence collection. So there's been a lot of research on determining who sheds epithelial cells or touch DNA. And there's been a little bit of conflicting research showing that is someone always a heavy shedder, or is someone always a light shedder? Are there consistently high or low shedders, or can there be variability? And we do have some conflicting research of showing that someone might just be a high shedder, but if they're tested again later, maybe they have transferred more into the low skin shedders. So there has been some variability in categorizing high shedders or light shedders. Overall, males have been found to be higher shedders than females, specifically young adult males. So between the ages of 18 and 45, they seem to have the higher rates of shedding than females of the same age. So when you think about, all right, oftentimes who are our suspects in these sexual assault cases? They oftentimes are young adult males, 18 to 45 years, which might be their shedding prime, so to speak, of their life, as they are higher shedders during that time period. So what else impacts donor shedder status? Well, hand washing. No huge surprise there. There's found that if somebody has recently washed their hands, there's a decrease in touch DNA, very likely because that washing of the hands, those potential shedded skin cells that are on the outside get washed away along with dirt and germs. But there also has been some other studies that have shown that effect may be not very long lasting, that it may be just right after the hand washing, but then it's not going to impact the deposition of touch DNA. It has been found that individuals who touch body parts or their face with their hands have an increase in touch DNA. And this makes sense, right? Because then they're not only having the skin cells from their hands, they are actually collecting skin cells from other parts of their body that are then on their hands. Another condition, or two conditions found to impact shedding status, are sweaty hands and dry hands. Those may sound like opposite, but let me explain why. When someone has sweaty hands, that sweat can carry DNA that then can transfer along with those skin cells to objects or other people's bodies. Dry hands, there's just more flaking off of that skin, so also has higher rates of shedding. Another big impact on the development of touch DNA is the surface of the object. So porous substrates like wood or fabric, they're going to have a higher yield of touch DNA than non-porous substrates such as glass. So let's think back to that case study. That smooth metal button, now the victim was touching it, which her friction of her fingers fiddling with that button removed that foreign contributor DNA. But even if she hadn't been touching that, potentially we would have had less DNA recovery on that smooth metal button than that lacy panel of the underwear. So the lacy panel of the underwear was a rough texture. That rough texture likely held on to those epithelial cells that touch DNA even more so. So then we have to consider, is skin or clothing more likely to yield foreign contributor DNA? Well, one study has shown that clothing is more likely. Very likely this is due again to the fact that clothing is going to be more of a porous substrate, a fabric, than skin. We don't have research confirming this. So this is Julie Valentine's opinion. But when I am doing an exam on a patient for a sexual assault medical forensic exam, I think about, when I'm collecting, even on skin, what is rougher textures that might hold on to that DNA more. So for example, we have a great yield of DNA profiles from neck swabs. When you think about it, a lot of people, especially as we age, neck gets a little wrinkly. There's some fine lines in that, little bit of a rougher texture. if I have a victim that tells me that their breast was touched, I want to find out if it was breast and nipples. Nipples is a rougher skin texture than smooth skin of the breast. I'm going to make sure that I collect from the nipples. A lot of our patients we see shave their pubic hair. Well, that creates really frequently kind of a rougher texture on the genital region, which likely can hold on when you do these pubis to anus swabs, can hold on to that DNA. And we have a high development of DNA from those swabs. But clothing definitely needs to be collected in these cases because it very likely is going to hold on to that DNA longer than skin. Also, the type of contact. So if there is contact with increased pressure and especially friction, that's going to increase the DNA yield on the object. So if you have a victim that explains that they were grabbed on an arm and maybe they were struggling to get away and there was friction between the suspect's hand and their arm, you're really going to want to collect where that friction was. And again, the increased pressure is more likely to transfer DNA. So we also need to consider, and we need a lot more research on strangulation and handshake. We actually are doing a study on DNA yield in strangulation, not ready to share those results quite yet, but we are looking into that as well. This brings up a whole other category of transfer, right, with touch DNA. What about transfer of DNA from one person to an object to another person? I'm not going to delve into that on this, but there is other research, and that's something we always need to consider as well. So length of time between contact and evidence collection, recent research has shown that that is a significant factor. Especially these researchers found that if there was friction, and they were looking at clothing, so if there was friction on that clothing item, that as time went on and there was an increased friction, there was a decrease in touch DNA yield. So they looked at the front part of a t-shirt, and then they looked at the buttocks region and found that if somebody's sitting down, when you sit down, there's friction created and you actually lose that DNA, that is a significant factor. The length of time in the National Best Practices for Sexual Assault Kits, a multidisciplinary approach, recommends for touch DNA collect within 72 hours. Now, we need more research in this to really inform this practice, but I think for now that the best approach is when in doubt, collect. So we believe that up to 72 hours, and we need more research to validate that time structure. So more research will improve our understanding of touch DNA. All right, so let me share with you what we have done in the state of Utah after this case happened, and I'm going to go into a little bit of our research findings on some other cases that are groping sexual assaults that supports these changes that we've made. One is we have developed a stranger touch DNA documentation form. But again, I want to be really clear. Now we call this more the touch DNA documentation form. This can be if there's a protective order. It also could be if it's not a stranger, but if you have a victim who is out on a date and maybe the kissing was consensual, but then this isn't a stranger but an acquaintance that puts the suspect puts their hand down the victim's pants. Well, we should be collecting touch DNA where that suspect's hand that was non-consensual, that contact. So you see we have here to list the item of clothing, and then it's really, really important to note the location and type of touch. I'm going to go back to that case study where we talked about the underwear. So the SANE collected the swab exactly where the victim said he touched the front part, this lacy part, the front panel of the underwear. The SANE did not write that information. Again, the SANE was sort of grasping at straws a little bit. Let's see if we're going to get anything at all. We have a violent perpetrator who has had multiple attacks in one day, and but did not write and share that information with the forensic scientists in the sexual assault kit. So the forensic scientists tested where they usually test an underwear, which is the crotch. So we try to be very specific on this touch DNA form to write where the victim was touched and to draw this out on the body diagram. We then have directions for how to fold and place the clothing. Now, when we have educated law enforcement on this, we've shared that they can go ahead and collect touch DNA with this form. We really prefer, strongly prefer, for them to call the forensic nurses. One, our primary objective is to help the victim heal. We never want to diminish the trauma that happens from these groping sexual assault cases. And when you have a nurse who is trauma informed, to provide resources, help that victim begin to heal, really, really important. And also then to sometimes we find out, oh, maybe more private areas have been touched. But we do try to collect the clothing if the victim is willing. So again, application to sexual assault, definitely groping sexual assaults by strangers, groping sexual assaults by known individuals, depending on the body location, as I discussed, sexual assault by strangers. So I had cared for a patient who was sexually assaulted, penetrated by one individual, penis and vagina. Another individual had held down her parts of her body. So I swabbed for touch DNA where that victim had told me she'd been held down by that second suspect. Sexual assault by multiple perpetrators goes into that as well. Find out where all perpetrators have touched. And then assailant evidence collection for touch DNA of the victim. You may have other ideas that you think we need to expand our thinking about where we collect touch DNA. So this is applying touch DNA background to practice. This is looking at our research on groping, just groping sexual assault cases. We're going to talk about victim and assault characteristics and the DNA analysis findings. So this is a retrospective review from 2010 to 2017, a little over 4,000 cases. Across most of the population in Utah, it represents 78% of the population. The information I'm sharing with you are all cases where the victim was over the age of 14. They had a fully collected sexual assault kit with evidence collected, and they wanted to report to law enforcement. So there are no restricted cases in this. We collect, our research team at this time, we now collect over 280 variables, but at this time it was 241 variables per case. So what our question was is, are there differences? Our first question was, are there differences in the victim variables between groping sexual assault cases and penetrative sexual assault cases? And we looked at a variety of factors. You can see here are those that we found not to be statistically significant difference: age, gender, race, a patient or suspect use of drug or alcohol, if it was a multiple assailant assault, and if it was suspected drug facilitated assault. But we did find some differences. We found statistically significant differences on the assailant-victim relationship, the location of the assault, if a weapon wasn't involved, and if the assailant was injured or possibly injured by victim's actions, which means the victim fought back more during the assault. So on our first finding, We found the assailant and victim relationship in groping versus penetrative acts were more likely to be committed by stranger, 30% to 18%. And I'm going to tell you how we define stranger. We define stranger as the victim does not know the perpetrator's name and has not had a meaningful experience with them. So they may have been at a party or seen the perpetrator at some event, a bar, on campus, wherever it might be, but have not interacted with them, nor do they know their name. So that is 30%. We also have those stranger cases, right, of jumping out of the bushes, stranger cases like what I shared with you on our case study where this victim was attacked by a stranger in a parking lot. Current or ex-partner, spouse sexual assaults are 14%. So domestic violence sexual assaults are 14% in our penetrative acts, but 6%, so less, in the groping sexual assaults. Interestingly, we found that the category of other on the assailant or suspect, which we categorize oftentimes, it's generally someone who's in a position of authority. So it might be a boss, a manager, a teacher, a bus driver, that those victim assailant relationship, when it's somebody who's other or position of authority, much higher in the groping sexual assault cases, 11% to 5% compared to penetrative sexual assault cases. We also looked at location of assault. We found in groping cases, less likely to happen in a house or apartment, but much more likely to happen in a car, 20% compared to 8%, outside, 19% compared to 10%, and other. And our other category for location, our number one is motel, but that's also a bar, a restaurant, and other, a building or facility, place of employment, which aligns very much so with our category of other in relationship, that it's somebody who is in a position of authority. So victim action, again, we found that if the assailant was injured or unknown if injured, 79% compared to 53%. We found higher weapon use in the groping sexual assault cases, 15% to 10%. So out of these 66 cases that we found that were groping sexual assaults, no bodily fluids, 31 of the sexual assault kits were submitted to the state crime laboratory. We passed legislation in 2017 in Utah mandating the submission and testing of all kits. But again, these were cases from 2010 to 2016, so only 31 of them were submitted. Out of those 31, quant male DNA was found in 84%, or 26 of those sexual assault kits. Out of those, half of them, 13, had enough quant male DNA found to progress to STR DNA testing. For those 13 that had STR DNA testing, 10 out of 13, or 77%, developed single profile probable STR DNA profiles of the assailant. 3 out of 13 developed no comparable data. But that's pretty astounding that 77% of those that went forward developed a probative profile. We also looked at YSTR testing, and 7 of the sexual assault kits also had YSTR testing, and 6 of the 7 developed a single profile that was the probable assailant. So every time you do research, you have to consider, so what does this mean? And now what do we do with this? Well, we believe this is what this means, that DNA analysis findings support collection of DNA evidence in groping sexual assault kits, also supports the sexual assault kit submission of DNA evidence that are collected in these kits, and the DNA analysis findings support analysis of evidence collected in groping sexual assault cases. So the bottom line, we should all be working together to collect DNA in touch DNA in sexual assault cases. So we need to collect, we need to submit, and we need to analyze. So then we have the whole issue, and I'm going to quickly go over this. We need a lot more research on, for touch DNA, when we are looking at a smaller quantity of potential cells, what matters? Swab type. Do you collect with sterile water or other solution like a detergent? How many swabs? What part of the swabs? All of these conversations are really important that jurisdictions have between the forensic medical examiners, or SANES, and the forensic scientists. Communication is absolutely key. So I referenced before the national best practices for sexual assault kits, recommendation 8. And for those of you who work in this field, I hope you read this, the national best practices, frequently and refer to it. But recommendation 8 says examiners should concentrate the collection by using no more than 2 swabs per collection area to not dilute the biological sample. And if one swab is collected, that they need to be collected consistently. So please refer to this document. Again, here is an exact screenshot from page 21 that talks about collection, and you'll notice it says same for children as adults. We also, when we are looking at having our best sample possible, we have to consider avoiding cross-contamination. So of course, single-use tools and lubricants. We want to limit the number of people in the room, a support person at a distance, glove and mask worn, avoid talking. If there are any males in the room, especially, make sure they are wearing a mask and are located far away from those collected swabs. This is just a shot of what our, in Utah, our sexual assault medical forensic exam form looks like. You'll notice that we have on skin swabs, we have A, S, and E. The forensic examiners or SANES indicate if they have collected from that location for saliva, amylase, seminal fluid, or epithelial cells. And that helps to guide for the forensic scientists. So there's a whole bunch on this front about what does the future hold. There's research out there looking at, and this is no commercial endorsements, mini tapes have been found to be quite effective in collecting touch DNA, especially from clothing. On this presentation, I have a number of references to look at. And then educate, especially with law enforcement. We've had a focus on campus police and transportation police, because we have seen that frequently those might be the jurisdictions, and consider airports, because what happens on planes regarding sexual assault, those might be the law enforcement agencies where we see more groping sexual assaults. So I hope that you take this information, go back to your multidisciplinary teams, share this information. We want to see these victims of groping sexual assault cases collect evidence, provide trauma-informed care. Takeaways, we should collect this, and also we need to think about where touch DNA evidence in groping sexual assault and other sexual assault cases should be collected. With this webinar, our State of Utah Touch DNA form will be uploaded. We have a full article that's an open access article. We also have archived webinar and an additional webinar on sexual assault cases and videos. And with that, I will take questions. I will just quickly show you our references. Thank you.
Hi, everyone. I'm Michaela Askley, the forensic scientist at RTI International. I'll be your moderator. Thank you, Julie, for joining us for another webinar in this series. we'll go ahead and start the Q&A. Looks like there's quite a few questions related to that first case study. The first question reads, was the victim first discussed unable to scratch the perpetrator?
The victim did not report scratching the perpetrator.
Thank you. All right. Another question related to the scene, was the vehicle in which the incident occurred processed?
I do not believe the vehicle was processed at that time. This was in 2011. Things may change now if that had happened, but I do not believe that vehicle was processed.
Sure. I know different jurisdictions always do things differently as well.
Correct.
All right. Another question is, do you know how much time passed between the sexual assault exam and the actual assault?
Yes, that was pretty speedy. What happened is the victim, when her car alarm went off, which we're really not quite sure how that happened, but a true blessing, and the perpetrator ran away. In all of the struggle, her contacts had been knocked out, and she closed the door and started driving, was able to find her phone and called 911, but was not able to see very well. Kind of knew the general area where she was. And so a campus police car was dispatched and found her, took her to the station. So I believe the exam happened probably between 2 and 3 hours after the assault.
Thank you. All right, another question related to that first case. When the underwear was examined, did they only test the crotch area? If they would have just collected the underwear, would the nurse need to indicate that the underwear were touched in a specific place?
Yes, and that was, this case was a real learning case for us in Utah. The forensic nurse did not document where on the underwear it had been touched to the forensic scientist. The forensic nurse knew that information because the victim had told her that information, but didn't document it. Now our touch DNA exam form very clearly lists to the forensic examiner to document where the touch had occurred on the clothing.
Great, thank you. And then what was the method of collection? So one wet, one dry swab, what kind of swabs, cotton tipped, were used, and how was it packaged and stored?
Yeah, great question. So our guidelines in our state, we do not do wet and dry, one wet and one dry. If in your jurisdiction you do that, just make sure to label, this was wet one done first, dry one done second. In our jurisdiction, we do 2 swabs at the same time, cotton tip swabs, moistened with sterile water, and then swab the area. We same as everywhere else, we then leave them to dry and package them in our boxes and then seal them in an envelope in our sexual assault kits. There has been some research looking at, rather than moistening with sterile water, there's some commercial detergents on the market, moistening cotton tip swabs with detergents that I think look really promising. I think that would be an exciting area for continued research, especially when we're looking at touch DNA, which is in that kind of umbrella of trace DNA, smaller amounts of DNA, but specifically for touch DNA from epithelial cells.
Right, great, thank you. Here's an interesting question, but in relation to hand washing and the shedding discussion, what about the effect of pre-COVID versus post-COVID?
Well, that just assumes that we think people are washing their hands more, right? But we don't know that for sure. When we think about a perpetrator acting out and committing an act of groping or sexual assault. That perpetrator may not be considering washing their hands before and maybe nervous, and so their hands might be sweaty, which sweaty hands have been shown to have an increased yield of DNA. So I don't have any information on that pre and post COVID, but that would be making an assumption that people would be washing their hands more, which I'm not sure if that's happening or not.
All right, and then another question here. Would a victim's hair be a viable source of touch DNA? This person has said, I have had victims whose hair was grabbed and held by the suspect during the sexual assault.
Oh boy, I have never gotten that question, and that is an incredibly intriguing question. I would say, when in doubt, collect. I have collected victims' hair before. I've had a couple cases where there's been semen, that you actually could see some dried semen in the hair. That's an obvious thing that you're going to cut and collect. But I don't know the answer to that. I would say that if you are doubtful, collect some strands, package that up, and submit that with the kit with a note to the forensic scientist. This is why I collected. The victim stated that because when you think of holding hair, that's probably a pretty firm grasp, and there's probably some friction also with the hair moving, which potentially could lead to some DNA, but I don't know. I think that'd be really interesting to look at.
Yeah, agreed. And kind of a follow-up to that, you're saying, when in doubt, to collect. We have a question asking, what are your thoughts on nurses not collecting from locations that are specifically stated per verbal history from the victim? This appears to be because the sample would be a touch-only sample.
Michaela, will you repeat that question?
Yeah, of course.
I'm just looking for it too.
Yeah, so they're just asking your thoughts on nurses not collecting from locations that are specifically stated. So I assume if the victim is, stating where they were touched, and then nurses not collecting from that area.
Yeah. I think, and again, it's going to depend a bit on the relationship, right? If you have a victim that you're seeing that reports that their arm or hand was held and they were out on a date with the assailant. I do not think collecting from the hand or arm would be very beneficial, because that was the victim stated that was a consensual act, right? If it's a stranger, or if there's supposed to be a no contact or a protection order, finding any DNA from that assailant could potentially be useful. So in those situations, I would collect in the touch areas. If a victim said it was touch only of their genital area and it was non-consensual, or not bodily fluids, but it was touch only that was non-consensual, I would definitely collect from there.
Okay, great, thank you. Hopefully that clarified for the person who asked that question. All right, another question, kind of dealing with at the scene. Since we're dealing with touch DNA, would it be better to have the victim processed on scene instead of transported to a hospital for the same to perform, since vehicles can present additional DNA?
So our guidelines are generally, we do our exams in hospital settings. This case study that I provided, the victim did not want to go to the hospital, but that's an unusual setting for us to do that in a law enforcement agency. Regarding doing that on scene, I think, you first have to consider the safety of all involved, including the forensic examiner. And on scene may not be very conducive to, sometimes we need to collect swabs from, intimate private areas. And I think that a victim would be more comfortable in a room or someplace where they had more privacy. So I think the on scene regarding if their law enforcement is collecting from a car, that would be on scene for them. But the forensic examiner, I think it would still be better to do that where they could sit quietly, provide trauma-informed care to that victim, because these are very traumatic experiences also. So it's meeting the mental health needs of the victim as well, which on scene could be a little chaotic. Sometimes getting away from there might be better. Regarding EMS collecting these, we haven't broached that at all. They generally will transport the victim as needed to a safer care environment.
I love how you stress the importance of trauma-informed care.
Absolutely.
All right, another question reads, what is your state's conviction rate and how many kits do you have backlogged due to insufficient funding for testing?
So I've got good news and bad news there. The good news is that we currently do not have a backlog. Our state has had a few SACI grants and a Danny grant, and we have cleared our backlog, which is great news. We also passed legislation in 2017 that mandates the submission and testing of all kits. So we've gone from 38% submitted kits across the state to now about 100% submitted kits, and we've gotten rid of our backlog. Regarding prosecution, I am talking about that next week for that webinar, so join in then if you would like. Just like most areas of the United States, our prosecution rates are poor. They are low, and there's many factors that we're going to be discussing next week on this. I think, this is an issue every part of the country. I think we have seen these major improvements on submitting sexual assault kits, but then our prosecution rates have stayed fairly stagnant. Huge area we need to look at. So join us next week.
Great. All right, everyone. I just want to mention to everyone still with us, we're going to go over a few minutes just to give Dr. Valentine a chance to answer a few more questions. I know, again, we're at the top of the hour, but for the next question, for DNA collection, when should one use MVAC over swabbing or scraping?
Yeah, MVAC. So MVAC, again, no commercial endorsements at all. MVAC is a way of kind of like vacuum collecting DNA. This definitely needs to be something you would need to talk to your crime lab about. Some law enforcement agencies actually have the MVAC machine. So it just needs to be a communication with your crime lab if they process that evidence that is collected from DNA. One of the issues whenever we're looking at small amounts of DNA is frequently you'll get more than one profile, right? So you're looking for any foreign contributor profile. When you're looking at these smaller amounts, and this happens with MVAC or other methods where you're looking at smaller amounts, you may get a higher amount of multiple profiles, which can be a bit more challenging on the interpretation on the forensic scientist's part.
For sure. And then in terms of this study, what is the DNA extraction method that was used? So MVAC, scrape, or swab?
So this was all from swabs, the information I shared with you.
Okay, thank you. All right, we have another question from the audience. In your review of 4,000 cases, were there any findings specifying the completeness of the STR profiles obtained?
So the STR profiles obtained that I shared on this, these were all partial enough to go into. So when we think of CODIS, right, we have a state database of CODIS and then a national database, and national database is more discriminatory. So the STR profiles that were obtained were enough of a partial profile that it could have gone into at least the state CODIS database.
And I think that kind of follows up this next question, was asking, was the genetic profile from the evidence uploaded and searched in CODIS? So,
yes.
All right. Another question asks, is there a reason why the dry and wet swab need to be differentiated?
Yes. So I was on the committee. It was called the SAFER Committee, Sexual Assault Forensic Evidence Reporting Act, working group that developed the National Best Practice Guidelines that were published in 2017 by NIJ. And this is actually a topic we discussed a lot, and we had many expert forensic scientists that were part of this working group. They felt that it was really important to differentiate if two swabs are collected at different times, whether they be wet, or whether they be, you do wet first and then dry, to share that information with the forensic scientists, that this was the wet swab, this was the dry swab. That the more communication as those of us that collect the evidence that we can share with those who analyze the evidence, the better. So this is, again, just an area where you are sharing information. This was the wet swab, this was the dry swab. Jurisdictions may look at those differently at the individual crime labs, just like you want to share, I am collecting these swabs from the breast that was touched, so that they have an idea, oh, probably looking at trace DNA here, this was touch DNA, what are we going to find? Again, just improving that communication.
Absolutely. All right, this should be a quick question, but someone's asking if you have an estimated date of publication for that strangulation study.
Oh, that's a really good question. So I had a graduate student who defended 2 weeks ago. This a portion of this study was her graduate work. And so we are probably going to submit that study in June. And your guess is as good as mine. Sometimes it takes a while to get these published, but we have a huge amount of cases in the thousands. And so we are really excited to get this publication out there. We have some really interesting findings.
Yeah, that's great. All right. Well, thank you for everyone for sticking with this webinar. And I'm afraid we won't be able to get to all the questions today. If your question wasn't answered, feel free to reach out to Dr. Valentine. But thank you so much for staying a little extra for those questions. And thank you all for being here.
Thank you.
Before you leave today's webinar, I'd like to draw your attention to our feedback form. It takes less than 5 minutes to complete and can be accessed via the checkbox icon directly above the chat. Your honest feedback is invaluable as we continually search for ways to improve and provide the best experience possible. Lastly, across the top of the live stream window are links to our social media profiles, including Twitter, LinkedIn, and Facebook. There is also a link to the FTCOE website, the Just Science podcast, and the FTCOE newsletter registration. Like, follow, or subscribe to stay up to date with everything the FTCOE is doing in support of the forensic community. Thank you again for attending, and we hope everyone has a great day.